Good afternoon, dear students. Today, in a series of microbiology lecture in SANDHAN series, we are bringing to you a very interesting topic, that is microbial nutrition. હવે આ ટોપિકના ડિસ્કશન માટે આજે પેનલ ઉપર છે on my extreme left Dr. Rakesh Panchal from MB Patel Science College, Anand, અને મારી બિલકુલ અડીને બાજુમાં જે બેઠા છે એ ભાઈ છે કિરણભાઈ રાજપૂત. એ પણ આણંદથી જ MB પટેલ સાયન્સ કોલેજમાંથી જ છે. બંને તજજ્ઞોનો જે એક્સપિરિયન્સ છે ખૂબ વધારે છે ટીચિંગની અંદર અને સારા ટીચર્સ તરીકે નામ કમાયેલું છે. હવે આ ટોપિકની હું સહેજ આછી રૂપરેખા આપી દઉં કે વોટ ઇઝ માઇક્રોબિયલ ન્યુટ્રિશન. Microbial nutrition basically it is a kind of a nutrition requirement of microorganisms. Microorganism ने जे पोषक तत्वों एमना सेलना सर्वाइवल माटे जे जोईए छे ए पोषण ने आपणे कहीशुं माइक्रोबियल न्यूट्रिशन. For any cell, if it has to grow, it has to survive and it has to reproduce, it requires two important things. One is energy and the basic elements for cellular build-up. એક તો એનર્જી જોઈએ જે સેલના રૂટીન વપરાશ માટે જરૂરી છે, સેલની એક્ટિવિટી માટે જરૂરી છે, અને બીજું કે સેલના કોમ્પોનન્ટ્સ માટે જે કંઈ પણ એલિમેન્ટ્સ જોઈતા હોય એની જરૂરિયાત આ ન્યુટ્રિશન પૂરું પાડે. Now this is not only true with microorganisms alone, it is true for any living cell. ગમે તે પ્રકારનો સેલ હોય, એ પછી માણસનો સેલ હોય, પ્લાન્ટનો હોય, એનિમલ્સનો હોય કે બેક્ટેરિયાનો હોય, દરેક માટે એ વિધાન એટલું જ સાચું પડે છે. But today we are going to restrict ourselves mainly to the understanding of microbial nutrition. How organisms they derive or they obtained their nutrients, their various elements from the surroundings, and on the basis of that, how do these organisms they carry out their routine work. On the basis of the nutrition which is consumed by microorganisms, which is being utilized by the microorganisms, organisms ને આપણે જુદી જુદી રીતે ક્લાસિફિકેશન પણ કરી શકીએ છીએ અને એ બધું પણ આપણે ડિટેલમાં જોઈશું. આપણે એ પણ જોઈશું કે ઓર્ગેનિઝમ્સ જે છે એ રૂટીન ઓર્ગેનિઝમ્સ કેવી રીતે પોતાનું ન્યુટ્રિશનલ અપટેક કરે છે અને એ સિવાય અમુક એવા ઓર્ગેનિઝમ્સ પણ છે કે જે કાં તો ફેસ્ટિડિયસ છે કે જેને સ્પેશિયલાઇઝ ટાઈપનું ન્યુટ્રિશન જોઈએ છે, અથવા તો અમુક ઓર્ગેનિઝમ્સ જે એક્સ્ટ્રીમ એન્વાયરમેન્ટ્સમાં રહે છે. એક્સ્ટ્રીમ એન્વાયરમેન્ટ્સ કે એવું એન્વાયરમેન્ટ કે જ્યાં આગળ રૂટીન ઓર્ગેનિઝમ્સ સર્વાઈવ ના થઈ શકે, તો એમના માટે નેચરલી છે કે એ એક્સ્ટ્રીમ એન્વાયરમેન્ટનું રિક્વાયરમેન્ટ્સ, ન્યુટ્રિશનલ રિક્વાયરમેન્ટ્સ જુદા પ્રકારની હોય, કારણ કે એમને સર્વાઈવ થવાનું છે, એમનો સર્વાઈવલનો પ્રશ્ન છે. તો ત્યાં આગળ જે અવેલેબલ ન્યુટ્રિયન્ટ્સ છે એ રૂટીન ટાઈપના નથી હોતા, જે નોર્મલ બીજા ઓર્ગેનિઝમ્સ યુઝ કરી શકે એવા. એટલે એમનું પણ જે ન્યુટ્રિશનલ રિક્વાયરમેન્ટ છે એ ખાસ જુદી હોય છે. તો આ બધું ડિસ્કશન સાથે લઈને આવ્યા છે આપણી સાથે રાકેશભાઈ અને કિરણભાઈ MB પટેલ સાયન્સ કોલેજમાંથી. તો હવે આ સબ્જેક્ટનું ડિટેલ ડિસ્કશન પહેલા શરૂઆત કરશે ડોક્ટર રાકેશ પંચાલ. પ્લીઝ રાકેશભાઈ.
Thank you, Rakeshbhai. Nutrients are matter which has been thought for years. When we see a skeletal body of a person, we just see that what the person will do. normally the function of person may be having some problem. Is it so with microbes? If the nutrients are not proper, then microbial function that is been disturbed. Why we are concerned with nutrients of microbes? The reason is numbers of microbes we are using for commercial activity. Numbers of microbes are causing disease to us. Numbers of microbes are harmful as well as useful to us. So we need to grow them. When we need to grow them, we need to know about their requirements. We need to know which elements are required by them. We need to know that what is the environment in which they are habituated to grow. And all these things sum up in form of a kind of nutrients required for the growth. If you go for classical definition of nutrients, then as per the Webster, something that nourishes or promotes the growth and repair the natural wastage. That has been termed as nutrients, particularly for organic life. Microbial life is single cellular. More specifically, if I say nutrients are nothing but the elements and combination of elements. More number of elements are combined which are specific requirement of a particular organism, and for the growth as well as repair, the things are required. The nutrient has been classified differently, as we see that here in the presentation, that the nutrients are of basic two types required by microbes. One is the source of energy, and the other one is the raw material which need to form different cell components. The raw material which are required here are some of the precursors which forms for anabolic activity, and the energy source required that may go for catabolism, breakdown of energy, lot of energy released that has been used by precursors, and ultimately the anabolic activity where the cell constituent are been formed, and ultimately cell may grow. When we go for nutrients, we divide into two major types. One is organic nutrient, another one is inorganic one. For organic nutrient, it's nothing but the debris of the living creatures, plants, animals, and microbes, which is rich in organic substrates. By and large, it contains the carbon, hydrogen, and oxygen, which is a tremendous supply. And where we go for the simplest one, that is glucose, and to more complex polymers like cellulose, lignin, chitin, all this kind of organic substrate, which may be required by microorganisms for their growth. In inorganic nutrients, they are other than carbon and hydrogen. Normally, inorganic nutrients are available in mineral deposits, that is in earth crust. Similarly, tremendous amount of inorganic salts are there in water body. Very nice example is seawater, tremendous amount of sodium, that is a kind of reserve there. Another one is available in atmospheric air in form of various gases. Metal and their salts like magnesium, sulfate, ferric nitrate, sodium phosphate, etc. are the key requirement for the growth of microorganisms. Among gases, it requires oxygen, carbon dioxide, and water, which are been there available in atmospheric air. If you go for the cell content of microorganism, particularly in bacterial cell, then 80% of the weight of cell, that is water. Remaining, if you go for the dry weight of cell, where the water is lost, we'll find that 40 to 70% protein, 13 to 34% nucleic acid, and 10 to 15% lipid. Trace amount of the monomers, intermediates, and inorganic ions. The things which are there in bacterial cell, the things may be required to be added in form of medium if you want to grow the organism like. If you go for the content wise, carbon, oxygen, nitrogen, hydrogen, phosphorus and sulfur, these are the major nutrients required for the growth of microorganism. And among them, the nutrients available or the amount available in microbial cell, if you go up for dry weight basis, you will find that 50% of dry weight of cell is carbon, 20% of it is oxygen, 14% nitrogen and hydrogen, 8% phosphorus, 3%, and remaining is sulfur and potassium. The thing which is there in cell probably will have to provide the same thing in form of medium. So while formulating medium or while providing the nutrient to microorganism, we'll have to keep in mind that the things which is there inside the cell, the according proportion should be required by the cell for the growth of its own, and hence we'll have to formulate accordingly. If you go for this carbon, which is normally used for the structural constituent of the cell, whereas oxygen normally present in all the major molecules which are there inside the cell, as well as oxygen is the best electron acceptor which is needed by the organisms. Nitrogen normally used to be there in nucleic acids, then nucleotides in form of coenzymes required by the cell. The hydrogen is required, that is the main constituent. As you know that carbon, hydrogen, oxygen, these three are the main constituent required for the cell for its growth, and the component which is normally present in water. Phosphorus, that's constituent of nucleic acids, nucleotides, phospholipids and teichoic acid. Similarly, sulfur is required as a constituent of the sulfur containing amino acid, along with cysteine, cysteine, methionine, that is there. Sulfur is functioning as excellent coenzyme also, so sulfur requirement is there. Potassium, similarly main cellular inorganic content, that is potassium, it functions as cofactor in certain enzymes. Magnesium similarly used as cofactor. Calcium, that is excellently required for the strengthening of the endospore body, so that is also needed by majority of organisms. Iron is required in form of different inorganic and organic, that is the salts basis, and it required for kind of heme, that is the major requirement for microorganism for their growth. The macronutrients, as we know, that is the nutrient which supplies major cell structure. Normally they are found as the components of carbohydrates, lipids, proteins and nucleic acids, which are the major content present in the cell. They are obtained from organic as well as inorganic substrate. The other one is micronutrients, which are required in trace amount, little amount. These micronutrients, they are manganese, zinc, cobalt, molybdenum, nickel, boron and copper. Normally their function is to take part as a cofactor in various enzyme activity, so they are having its key role. Though it requires in little quantity, the role is major because the functioning of enzyme that is based upon these coenzymes. Now we'll go for macronutrients. Among it, the carbon, that is the first and prominent requirement, that is the skeleton or backbone of the cell, that is carbon. And this carbon, that is been supplied in form of organic carbon to the organisms, that is reduced preformed organic molecules which are product of other living organism like debris of plants, debris of animal and debris of other microorganisms. They can be simple to large polymers in form of glucose can be supplied, very simplest one. Another one is the polymer may be starch, cellulose, lignin, like polymers supplies carbon in form of organic. Inorganic carbon, when we see, this carbon dioxide is the most prominent and the only inorganic carbon which is to be supplied to microorganism, synthetic carbon source which has been used by microorganisms. For nitrogen, the nitrogen is required for synthesis of amino acids, that is the backbone of protein. Then nucleotides, purine pyrimidines, they are been formed from nitrogen, enzyme cofactors and other substrates. Among nitrogen also there are two major supplies. One is organic nitrogen, that is primarily obtained from the catabolism of amino acids and nucleotides which have been formed by nitrogen. The same can be degraded and can be utilized by microorganisms. Inorganic nitrogen, that is the oxidized form of nitrate and nitrite, can be supplied in form of medium to the organisms. Even reduced form of nitrogen, that is in form of ammonium ion, can be supplied to organism. And there are still certain microorganisms which has got capacity to utilize atmospheric nitrogen. Tremendous amount of nitrogen, around 72 to 74% is there in the atmospheric air, which can be used directly by this organism. Organism has got excellent enzyme system which can utilize this nitrogen, fix the nitrogen and convert into organic nitrogen. So that nitrogen source is an excellent source, particularly for the nitrogen fixing organisms. Oxygen requirements, oxygen is obtained in form of the molecular oxygen that is available in atmospheric air. Then component atoms of various nutrients, that is oxygen is combined with major organic molecules. So from there oxygen can be utilized, and oxygen can be utilized from water, because water is the basic material from where all the kind of nutrient is supplied. For sulfur, there used to be inorganic sulfur supplies in form of oxidized sulfate, SO4. Reduced inorganic sulfur may be kind of sulfide H2S. There used to be H2S utilizing microorganism which has got tremendous populations, and these are the organisms which are economically important also, as you know that these are the organisms which can degrade even concrete. Cement concrete monuments, even the old heritage monuments are being corroded by this sort of organism which has got capacity to utilize H2S or kind of reduced sulfur which is there in cement as well as in the rock. Organic sulfur, the prominent is amino acids and the other one is nucleotides. That is dead organic matter contain tremendous proteins which can be degraded in form of amino acid, and from amino acid the sulfur can be utilized, that is sulfur containing amino acid, cysteine, cysteine and methionine. Another one is elemental sulfur. Some organisms can utilize elemental sulfur also. The green sulfur bacteria, as it has been projected here in front of you, this is the excellent organism which can utilize direct sulfur from the crust. So far as phosphorus requirement is concerned, this is a major element which is there in the energy molecule, which is transient and which has been converted from one form to another, that is lower energy to higher energy. The phosphate, that is excellent molecule, usually supplied in form of inorganic phosphate derived from the phosphoric acid H3PO4, where the inorganic phosphate can be utilized by microorganisms, so you can direct supplies it. And many organisms can utilize organic phosphate also, that is a kind of nucleotides can be utilized directly, degraded nucleotides, and phosphate can be used from that. So there used to be various organisms which has got capacity to utilize phosphorus like. Remaining materials which are been the major elements still, Dr. Kiran Rajput will explain the further elements.
Okay. Dear friends, so we will see that next four major other elements. One is potassium. So potassium is required for the activity of number of enzymes, including those involved in protein synthesis, as well as potassium is also having an important role in membrane transport. And normally potassium is supplied in form of inorganic metal salts like dipotassium hydrogen phosphate, potassium dihydrogen phosphate, potassium chloride and many more. In this way, potassium is normally supplied in the medium for the growth of microorganisms. It normally constitutes about 1% of the cell dry weight. Next one is calcium requirement. Calcium is also a major element which is required or which constitutes about 0.5 gram % of a cell dry weight. Normally it will act as a coenzyme of part of enzyme. For example, amylases requires calcium ions for the stability, as well as bacteria requires calcium ions for formation of endospores. So these calcium ions are supplied normally as a inorganic salts in the medium. Normally calcium chloride, calcium carbonate, etc. Next requirement is magnesium. Magnesium serves as a cofactor for many enzymes, as well as it complexes with ATP. It also stabilizes ribosomes and cell membranes. So magnesium is a very important element. It constitutes about 0.5% of cell dry weight, and it is also normally supplied in form of inorganic salts like magnesium sulfates and many more. Next one is iron. Iron is normally supplied in form of ferrous and ferric, Fe+2 and Fe+3. Normally it will found as a part of cytochromes, cofactor of various enzymes, as well as electron carrying proteins in a cell. Normally it is supplied in form of ferrous sulfate, that is FeSO4, and ferric chloride, that is FeCl3. So many iron oxidizing bacteria in nature, they can utilize this. For example, Thiobacillus thioxidans, Thiobacillus ferroxidans, these are the bacteria which can obtain or which can degrade iron from an iron ore. Next is the important requirement, that is minerals or trace elements. Trace elements are the elements which are required in very small quantity, that is micro quantity, what we call in micrograms, and requirement is very important because normally minerals or trace elements will act as a part of enzymes, that is normally we called as cofactors. Here we can see zinc. Zinc can be a part of active site of many enzymes, but it is also involved in association of regulatory and catalytic subunits in E. coli aspartate carbamoyl transferase enzyme. Same way, manganese, it will also a part of many enzymes, as well as it catalyzes the transfer of phosphate groups in various metabolic reactions. Molybdenum, it is a part of enzyme nitrogenase in many bacteria, and that is why molybdenum plays an important role in nitrogen fixing organisms in nature, and that is how molybdenum is essential element for fixation of nitrogen in nature. Next is cobalt. Cobalt is a central molecule of vitamin B12, that is cyanocobalamin, and that is very essential. In absence of cobalt, organism cannot synthesize vitamin B12. For example, Propionibacterium shermani and Pseudomonas species of bacteria can synthesize vitamin B12, that is cyanocobalamin, in presence of cobalt. So this is what the role of minerals. Some special metal ions are there in nature which can be utilized, or which can some organisms can grow in presence of this. For example, red extreme halophiles, they are the bacteria, archaebacteria, which can grow in presence of sodium, and at least they require 12 to 15 gram% of NaCl concentration in the medium for their growth. Here we can see the photograph of red extreme halophiles that is shown in picture. Even you can see the red sea. That red color is because of the pigment carotenoids produced by these organisms, and that is why seawater is normally observed red in color. Next one is silicon. Diatom algae utilizes salicylic acid to construct that beautiful cell walls of silica or silicon polymer, and we can see the beautiful pictures of diatoms that has been constructed from silicon. So these are the metal ions, specially metal ions for which can be utilized. The other growth factor requirements, organic compounds which are required because they are essential for cell components or precursor of such components and cannot be synthesized by the organisms, these are called growth factors. There are three major classes of growth factors: 1, amino acids; 2, purines and pyrimidines; and 3, vitamins. So these are the organic compounds required in micro quantity for their metabolism. So these are the examples of few vitamins and microorganisms requiring these vitamins. For example, folic acid is required for the growth of Enterococcus faecalis, pantothenic acid is required for growth of Proteus morgani, and many more. So these are the essential vitamins required for the growth of the respective organisms. In absence of these growth factors, organisms may not grow, and that is why growth factors are to be supplied in the medium for growth of that particular microorganism. Last but not least, the most important constituent or nutrient requirement is water. all nutrients must be in a solution when we provide when we want to grow organisms in the medium. Bacterial organisms are particularly, they are not having a system for uptake, and that is why all nutrients must be in a soluble form before entering in a cell. As we know, water is the most polar compound present on this earth, and that is why it is the best solvent, and major components are soluble in water, as well as it provides resistance to the sudden change in temperature. And water is also a very good chemical reactant that is required for hydrolytic reactions. And that is why, as we have seen earlier, 80% of cell constituent is water, and majority of the microbial activities is being conducted in presence of water. So water is very much an important thing. So next, nutritional types of microbes will be taught by Dr. Pancha.
Thank you, Dr. Kiran. When we numbers of different nutrients are required to organisms, is there any system where we classify the organism on basis of nutrients requirement? Can we say that this particular organism requires this particular things, or can we classify them systematically on the basis of their requirement? Then the answer is yes. We have a way where we can classify the organism systematically in form of nutritional requirement of them. The first and prime criteria for this classification, that is on the basis of the energy requirement. The energy is crucial for all the living creature. Similarly for the microorganism also. The energy is required for various anabolic activities, where it goes for synthesis of protein, or it goes for replication of its DNA, it goes for transcription and formation of its RNA, various types of RNA. So major cell constituent to be formed where the energy is required. And this energy can be utilized from various substrates. Now, on the basis of energy requirement, we can classify the organism, the first type, that is called phototrophs. Phototrophs are the kind of organism which can utilize the radiant energy in form of solar radiation. The photon energy which can be utilized, the organism do possess a photosystem which has got capacity to absorb the kind of photon energy and convert this energy into chemical energy. And the excellent example, that is the cyanobacteria, which has been projected in front of you, that says different cyanobacteria varieties are there which utilize light energy and grow. Another type, that is chemotrophs. The other than phototrophs is chemotrophs, where these are the organisms which rely upon chemicals as a source of energy. Organism which can utilize chemical compounds, maybe a kind of carbohydrate or maybe a other kind of fat, can be degraded by the organism and energy is derived from that. All these organisms comes under the title of chemotrophs. The best example is your routine organism, Escherichia coli, or the Bacillus species, which day to day in microbial laboratory we used to grow. All these organisms are kind of chemotrophs. Another criteria for classification of organism, that is the source of electrons. Means electrons donor is very important. Here there are two types, that is lithotrophs are the kind of organism which can utilize reduced inorganic compounds as electron donors, and they are termed as lithotrophs. Another one is organotrophs, where the organism which can use organic compound as electron donors, and here the electron supply comes from the organic material. So litho and organo are the two types on the basis of the electron donors, and photo and chemo are the two types where on the basis of the requirement of the energy. Where you go for photolithotrophs, here two things are combined, that is the source of energy as well as source of electron donor. Here photolithotrophs are kind of organism which are phototrophic, so they rely upon radiant energy, and at the same time they can use inorganic compounds as their source of electrons. Say for example H2S can be utilized by this organism, as we have mentioned that these are the organisms which has got tremendous capacity to use a kind of inorganic sulfur, and H2S is converted into sulfur, and elemental sulfur can be cleaved off and electrons are being utilized. Chromatium ocani, that is the kind of organism which has been projected, that is utilizing the energy from H2S. That is a kind of photolithotrophs. Another one is photoorganotrophs. Here energy that comes from the radiant energy, that is photo, and the electron donor kind, that is the organic. Organic compounds, say for example succinate can donate the electron. So succinate itself convert into fumarate, and 2 of the electron that comes out, and which can be used by the organism for its cellular function. The excellent example in this case, that is the Rhodospirillum rubrum, the organism which is projected as a spiral shaped flagellated bacteria, which are using energy source as the light energy and electron donor as the organic substrate. Another one is CHEMOLITHOTROPHS. Here the energy that is relied upon the chemical other than radiant energy, and the electron donor that is kind of inorganic. Here CHEMOLITHOTROPHs are the organism which can utilize ammonium ion that is being used as electron donor, and ammonium is converted into nitrate, and ultimately the electron is derived from that, and the organism is termed as CHEMOLITHOTROPHs, so organic chemicals. And best example is Nitrosomonas species, Galeonella species, which is projected in front of you. These are the CHEMOLITHOTROPhic organism which has tremendous capacity to deal with ammonium ions. Organism utilizes it and converts into nitrate. All these organisms are farmer-friendly organisms. Another category is the CHEMOORGANOTROPHS. Here chemotrophs meaning they rely upon the chemical energy. Another one is organotrophs, meaning they are the organism which requires organic substrate to donate electron. And the best example here is the glucose, which is the excellent electron donor molecule, organic substrate. One molecule of glucose that yield 24 electron to the cell, and the normal organism which are routine in microbial laboratory, that is Pseudomonas, which can use the organic substrate as electron donor. So CHEMOORGANOTROPHs kind of Pseudomonas, Pseudoflava that is projected in front of you. Even other Pseudomonas are also the varieties. Different species of Pseudomonas can utilize the chemical energy and the organic electron donor. The rest of the classification, Dr. Kiran Rajput will explain you.
Nutritional types of microbes on the basis of carbon requirements. So carbon is the most important requirement. Many organisms in nature are there. They can utilize carbon dioxide as a sole source of carbon for assimilation of various cell components. So these organisms are known as autotrophs. Heterotrophs are the other organisms which can utilize organic compounds as their sole source of carbon. And the third category is known as MIXOTROPHS. They can obtain energy from inorganic electron donors, but obtain their most of the carbon requirement from organic compounds. So these are the 3 major categories of on the basis of carbon requirements. Autotrophs are the organisms what we called as CHEMOLITHOTROPhic bacteria, which can utilize CO2, that is carbon dioxide, as a sole source of carbon. They are termed as autotrophs. Particularly Nitrosomonas, which is seen in the picture, is a CHEMOLITHOTROPhic bacteria or autotrophic organisms. These type of autotrophic organisms can utilize energy from reduced carbon sources like hydrogen, hydrogen sulfide, elemental sulfur as electron donors. Autotrophic bacteria can be cultivated in a simplest medium that we called as defined medium or synthetic medium. The requirement is very simple. Mixture of inorganic compounds, inorganic salt is supplied, and they have tremendous capacity for synthesis because they can fix the atmospheric nitrogen. Here the medium is shown for the growth of Nitrosomonas europaea. Here ammonium chloride, magnesium, potassium, calcium, manganese, sodium, molybdenum, zinc, copper is supplied, and organism will utilize major carbon requirement or a sole carbon from atmosphere. So organisms can fix carbon dioxide and then they will grow. So autotrophs can be grown easily. Next, heterotrophs. Heterotrophs are the bacteria which can utilize organic carbon compound as a source and as termed as heterotrophs. Heterotrophs are immediate concern to us. the greater population of microbial population in our immediate environment is of heterotrophs. For example, whatever the microorganisms which are of economic importance, they all are generally heterotrophs. As well as organisms which is producing various diseases in human beings, animals, plants, these are also heterotrophs. For example, here pictures of few heterotrophs is been shown, that is Escherichia coli, normal flora of our intestine, Azospirillum brasiliensis, it's a normally present in soil, Salmonella typhi, which can cause disease typhoid, Proteus vulgaris, it is also a skin flora, Staphylococcus, skin flora, Lactobacillus acidophilus, which is a normal flora of curd. So these are the bacteria which are immediate concern to us. So normally heterotrophs are present in environment in ample amount, large amount, and they can be cultivated in two types of medium. Another heterotrophic bacteria, that is Burkholderia sapia. This is a most versatile organism which can utilize about more than 100 different types of carbon compounds. So these type of specific heterotrophs are there in nature which can utilize diverse range of compounds. Heterotrophs can be cultivated in defined medium as well as complex medium. Defined medium, as we have seen, it is having an exact composition, each and every component is known and weighed properly and then added in a medium. So here is the medium for growth of E. coli, defined medium for growth of E. coli. Next, complex medium, that is nutrient broth. Here natural components like beef extract, peptone is added in the medium. Exact composition of complex medium is not known, and that is why it is known as complex medium. Generally heterotrophs are grown in a complex medium. Next category, that is known as fastidious heterotrophs. Fastidious heterotrophs are the organisms which have specific nutrient requirement, that is specially vitamins and other growth promoting substance. In absence of this growth promoting substance, that is vitamins, these organisms cannot grow. So for growth of that particular fastidious heterotrophs, we have to supply these type of specific vitamins in the medium for growth of these organisms. For example, for cultivation of lactobacillus, we have to supply about 8 different types of vitamins in the medium for the growth of heterotrophs. So this is about the cultivation of heterotrophs. Now, MIXOTROPHS. MIXOTROPHs are the organisms known as CHEMOLITHOTROPHIC heterotrophs, where they can utilize both type of substances. They can obtain their energy from inorganic compounds. So inorganic compounds will act as electron donors and will obtain energy from it. And as a carbon is obtained from normally organic compounds. So these organisms are known as MIXOTROPHs. Example is Desulfovibrio desulfuricans as shown in picture. The other example is Begiatoa. It's a green sulfur bacteria. It can also act as a MIXOTROPHs. Next will be explained by Dr. Pancha.
Another category, then all these organisms where we go for the cultivation in our laboratory, that is obligate parasites. When we say this organism, these are the organisms which cannot be grown on synthetic or laboratory medium. Actually, these organisms are causing disease to animals or human beings, and their nutritional and physical requirements are not been understood properly yet, and they cannot grow on artificial medium in the laboratory. Means that we are trying all our best to find the way to grow them, but it's quite difficult, and many a time it is quite impossible to grow some organisms in laboratory. We call them as obligate parasites, and if you want to grow them, then we need experimental animals like mice, where we'll have to inject the organism in the animal, and animal itself is serving as the medium for us, where the organism will grow there, and then after we can utilize them. The Mycobacterium leprae, that is an excellent example of the obligate parasite, where the organism causing leprosy. Normally, there is no way where we can cultivate it in laboratory medium. Well, if you need to grow them, then the mice, where we'll have to infect mice by this organism, and the organism is growing in the body of mice, and then after you can utilize it. That is the organism causing leprosy. Another one is Treponema pallidum, which is causing syphilis to us or to human being. This organism is a kind of spiral bacteria. Same in case of Treponema pallidum, where it is obligate parasite, cannot be grown on laboratory medium, but still try are going on where we try to cultivate them on experimental animals. Another one is Leptospira. This is a typical organism which requires long chain fatty acid as a source of carbon, and due to this character the organism infects human being and the human cell which contain long chain fatty acid, where the organism can grow very well and cause disease to human being. The Leptospira is again dangerous organism. To grow them in laboratory, again it is quite difficult. Another one is Rickettsia, where the Rickettsia, again they are causing infection to human being. The projected slides shows some of the Rickettsia which are kind of gram-negative, short, raw type organisms which has got tremendous capacity to cause infection to human being. Again, growth is very difficult, but still the trials are going on where we can use blood, where RBC can be utilized as a source of nutrients to them, and the organism is growing on blood agar medium at some extent, but the prolonged growth that is not been obtained. So it's a kind of obligate parasite. Another one is Chlamydia. Same in case of Chlamydia also, that is human pathogen, can cause sexually transmitted disease, as well as they are the organism which are having the efficiency to infect, and it can cause blindness to human being. Same organism cannot be cultivated on laboratory medium. We use experimental animal for that, and the trials are going on to find a way how we can cultivate this kind of Chlamydia on laboratory media, but still it's quite difficult to grow them on laboratory media. So obligate parasites are the organism which cause disease to us, and the growth on laboratory media is quite impossible. Rather, we do not understand its nutritional requirement as well as physical requirement, and some cases where the nutritional requirement is defined and understood, but the physical requirement cannot be provided properly. Hence, the organisms are difficult to grow on laboratory media, where we'll have to rely upon the experimental animals, where we'll have to infect animals, and animal can be sacrificed for the growth of such organisms. With all this, the nutritional requirement, the references as we have referred for this, that is normal microbiology title, that is 5th edition of Pulsar, Chan and Craig. Another one is Prescott, that's again title microbiology, 5th edition, that is Prescott Hurley. Third one is the title microbial word, again 5th edition of Steiner, Edberg. And the last one is the Brock biology of microorganisms, that is by Medigam Mamti, Meritko, and the Parker J. All this reference, I would request Mr. R.J. Patel to sum up the talk.
Thank you, Dr. Panchal and Kiranbhai. આપણે જોયું બધાએ કે પંચાલ સાહેબે અને રાજપૂત સાહેબે ખૂબ સરસ રીતે માઇક્રોબિયલ ન્યુટ્રિશનનું પ્રેઝન્ટેશન કર્યું. હવે અહીંયા થોડાક પોઈન્ટ્સ હું આપને જણાવવા માંગીશ જે થોડા ઇન્ટરેસ્ટિંગ પણ છે કે ઓર્ગેનિઝમ્સ જે છે એ સર્વત્ર બધી જગ્યાએ વાપેલા છે. હવા, પાણી, ખોરાક, આપણી બોડીમાં, બોડીની સરફેસ ઉપર, everywhere there is a presence of microorganisms. To that extent that certain groups of organisms, they are able to get that nourishment even in a water which is as good as a distilled water. ઘણી વખત આપણે જોયું હશે કે જો પાણીનો ગ્લાસ જે પીવાલાયક પાણી છે, જે આમાં આપણે માનીએ છીએ કે ડિસ્ટિલ વોટર કે મિનરલ વોટર જેવું છે, એને તમે થોડા વખત માટે અનડિસ્ટર્બ રહેવા દો તો ઉપર એક પાતળી મેમ્બ્રેન જેવું થઈ જાય છે. તો એ મેમ્બ્રેન નોર્મલી અમુક બેક્ટેરિયાની બનેલી હોય છે. આ ખૂબ ધ્યાનથી જોશો તો જ તમને દેખાશે. આવા ઓર્ગેનિઝમ્સ, એમની એ કેપેસિટી છે કે એ વોટરની અંદર કે જેમાં આપણે એમ માનીએ છીએ કે કોઈ ન્યુટ્રિશન નથી, છતાં પણ એમાંથી કંઈક મેળવી અને ગ્રો થાય છે. આવા ઓર્ગેનિઝમ્સને આપણે ઓલિગોટ્રોફ્સ કહીશું. ઓલિગો મીન્સ a very small quantity of nutrients are there, but still they are able to obtain it from them. Then there is another group of organisms which are predators also. You will be surprised, but there is a bacteria like Dello vibrio. એનું નામ છે ડેલો વિબ્રિયો, કે જે પ્રિડેટર તરીકે કામ કરે છે અને એક બીજા બેક્ટેરિયાના સેલની અંદર જઈ મલ્ટીપ્લાય થાય છે, ગ્રો થાય છે અને એ સેલને મારે છે. તો પ્રિડેટર પ્રે રિલેશનશિપ ફક્ત હાયર ઓર્ગેનિઝમ્સમાં છે એવું નથી, એવું આ ટાઈપનું રિલેશન્સ માઇક્રો ઓર્ગેનિઝમ્સની અંદર પણ જોવા મળે છે. ત્રીજી વસ્તુ, એક પ્રોમિનન્ટ માઇક્રોબાયોલોજિસ્ટનું હું એક કોટ કરવા માંગીશ. Dr. Atlas, who was a great soil microbiologist also, and according to him, there is a microbial principle of or a principle of microbial infidelity. એમના મંતવ્ય મુજબ what he said was that there is no substance in the world which microorganisms cannot use that as their cellular constituent, for their cellular constituents and therefore energy. It means એવું કોઈ પણ સબસ્ટન્સ નથી કે જે બેક્ટેરિયા ન વાપરી શકે, provided you give them certain time and certain period for their growth. Like you must have come across certain names like Pseudomonas. Pseudomonas organisms અને એવા બીજા બે ત્રણ organisms કે જેની અંદર પ્લાઝમિડ્સ આવેલા છે. એ પ્લાઝમિડ્સની ઉપર જીન્સ હોય છે અને એ જીન્સ લગભગ એવા સબસ્ટન્સીસમાંથી એનર્જી અને ન્યુટ્રિશન મેળવે છે કે જે આપણે કલ્પના બહાર હોય. ફોર એક્ઝામ્પલ, પેટ્રોલ ઓર certain disinfectants or xylene or certain aromatic compounds which are basically harmful for other organisms, toxic to other organisms. જો ઓર્ગેનિઝમ્સને આની અંદર એડપ્ટ કરવાનો સમય મળે તો એ પોતે એવી એન્ઝાઇમ સિસ્ટમ ડેવલપ કરે છે જેથી કરીને આ પ્રકારના ઓર્ગેનિઝમ્સ પણ ત્યાં આગળ ગ્રો થઈ શકે છે. આ સિવાય બીજી એક વસ્તુ કહેવા માંગીશ કે જ્યારે આપણે ન્યુટ્રિશન વ્યવસ્થિત રીતે જાણી લઈએ કે આ બેક્ટેરિયાને આ પ્રકારની ન્યુટ્રિશન જોઈએ છે, that they require carbon, nitrogen, this and this, either in organic or inorganic form. The next step is basically, on the basis of this knowledge, we can formulate a medium. And when we can formulate a medium, it is possible to grow them in the laboratory. So medium is nothing else but it is a balanced formulation of nutrients required by microorganisms. Indirectly આપણે કહીએ કે સપોઝ આપણે જ્યારે જમવા બેસીએ છીએ ત્યારે જેટલા પ્રમાણમાં કાર્બન અને નાઇટ્રોજન સોર્સ, જે તે ફોર એક્ઝામ્પલ રોટલી અથવા તો દાળ જે પ્રોટીનનો સોર્સ છે, રોટલી જે કાર્બોહાઈડ્રેટનો સોર્સ છે, એટલા જ પ્રમાણમાં આપણે મીઠું નથી ખાતા. કારણ કે મીઠાની રિક્વાયરમેન્ટ્સ જેમાં સોડિયમ સોલ્ટ્સ હોય છે કે ક્લોરાઇડ્સ હોય છે એ ખૂબ ઓછા પ્રમાણમાં જરૂર છે, માટે આપણે એને શાકમાં અથવા તો રોટલીમાં ટેસ્ટ તરીકે નાખીએ છીએ. જ્યારે કાર્બોહાઈડ્રેટ્સ અને પ્રોટીન્સની રિક્વાયરમેન્ટ્સ વધારે છે તો આપણે એ ક્વોન્ટિટી આપણા થાળીમાં વધારે હોય છે. So when you know that which components are required in which proportion, this is how a formulation of a medium takes place. And when we are able to successfully formulate a medium, then we are able to grow them in the laboratory. However, it is also a very true fact that whatever microorganisms at present which we know or which the world knows, only say 10% of that microorganisms are known, and very few of them they are cultivable in the laboratory. Still, by large, there are so many organisms which are present surrounding to us, either in soil or in sewage or in other environments, where we have not been able to successfully grow them in the laboratory. Why? The reason is we do not know about their nutritional requirements. Therefore, the world of microorganism is very large, and to know that in detail, it requires a lots of time. Still, we have been able to know only little amount of organisms, little bit of organisms about it, and then if we are able to know more and more in detail about the nutritional requirement, we will be able to cultivate more and more of them. પંચાલ સાહેબ, આપને કંઈક છોકરાઓને જણાવવું હોય તો 2 મિનિટ આપણી પાસે છે હજી.
આભાર રાકેશભાઈ. જ્યારે રાકેશભાઈએ કહ્યું કે સુડોમોનાસ પ્યુટિડા excellent bacteria છે. It's an organism which can even degrade the oil. So I would like to focus a prime requirement in case of all kind of accidents in environment, where few months you have seen that there was a oil spill, large oil spill in ocean near Bombay, and people are worried about that what would happen to the aquatic life. So there we do have microbes, and the microbes are the only remedy in this kind of accidents, where the Pseudomonas putida, which is explained by Rakeshbhai here, that is the organism. If you spray this organism and tactfully handle them, then these are the organisms which can degrade all this oil, and at a faster rate, where no other method is employed, the microbes are at your remedy. Similarly, if there is a kind of radiation leakage, you may imagine what would happen when radiation leakage used to be there. So there also we have a kind of bacteria which is Staphylococcus radiodurans. These are the excellent cocci which has got capacity to absorb the radiation and can store radiation inside the cell. A large project, which is a kind of mega project on the research work on Staphylococcus radiodurans, is going on at MS University, Baroda, along with the association to BARC, Baba Atomic Research, Bombay. So there they work out this kind of bacteria. So any kind of problem comes to environment, the microbes are at remedy.
થેન્ક યુ પંચાલ સાહેબ. આપે બહુ સરસ જણાવ્યું કે માઇક્રો ઓર્ગેનિઝમ્સ ન્યુટ્રિશનલ રિક્વાયરમેન્ટ જાણવાથી આપણે પર્યાવરણને પણ થોડો બચાવી શકીએ છીએ, કે જેની અંદર ઓર્ગેનિઝમ્સ આવા જ્યારે ડિઝાસ્ટર થાય ત્યારે એનો યુઝ એમની ન્યુટ્રિશનલ રિક્વાયરમેન્ટ જાણીને આપણે કરી શકીએ છીએ. તો હું અત્યારે બંને સ્પીકર્સ જે મારી સાથે છે, ડૉક્ટર પંચાલ, ડૉક્ટર કિરણભાઈનો ખૂબ ખૂબ આભાર માનું છું. સાથે સાથે I would also like to thank the Department of Higher Education for providing us with, for providing the students as well as the staff the opportunity to listen to such interesting lectures. And these lectures are basically very much helpful to the students. Last but not the least, I would like to thank the staff members and other crew members of BISEC department who behind the camera have been working very hard to make this program a great success. Thank you very much.
Thank you very much.